Hello, everyone. Welcome to today's broadcast, How Polymer Expertise and Data Analytics Can Deliver Optimized Performance and Processes for Your Formulations. I'm Rita Peters, Editorial Director of Pharmaceutical Technology, and I'll be moderating today's event. We're pleased to bring you this webcast presented by Pharmaceutical Technology and sponsored by DuPont. I'd like to share a statement from our sponsor. DuPont aims to help their pharma customers with both everyday challenges and future solutions. Armed with more than 300 years of combined cellulosic experience across their portfolio and unrivaled vital expertise, DuPont scientists and engineers fuel cross-functional innovations that solve all types of formulation challenges. Their broad and unique portfolio, including leading brands like Methylcellulose and Avicel, are designed to deliver performance and cost advantages in various oral solid dosage forms. Couple that with robust manufacturing, quality, and regulatory expertise, and DuPont can help deliver a consistent, high-quality offering at the right time, every time. I have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit your question by typing it in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the window, or you can hover your mouse over the lower right-hand corner and drag the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing the presentation, just click on the question mark Help widget in the dock at the bottom of your presentation window. Now I'd like to welcome today's speakers. Dr. True Rogers is a senior research scientist for DuPont Nutrition and Biosciences Pharma Solutions, where he's responsible for developing and launching new polymeric excipient offerings for modified release oral drug delivery systems. Prior to his current role, he had spent his career both Dow and as a pharmacist in community practice and hospital settings. True's research interests include formulation and controlled delivery of actives of any type, the structure of property performance fundamentals impacting application performance, and in various life sciences. He's also spent significant time on particle engineering for drug solubilization and bioavailability enhancement. True holds a bachelor's degree in pharmacy and a doctorate in pharmaceutics from the University of Texas at Austin. Dr. Yeli Zhang is a technical service manager for DuPont Nutrition and Biosciences Pharma Solutions. Prior to her current role, she worked at FMC Health and Nutrition as a senior scientist in excipient product development and support, and later became North America Health Technical Manager, responsible for providing technical support to FMC's North America excipient market. Before joining FMC, she worked at National Starch and Chemical Company as a senior scientist in starch excipient and delivery system development. Dr. Zhang received a doctorate at Cornell University in polymer science and biomaterials. So welcome to both of our panelists. True, I'm going to turn the presentation over to you.
All right, so Rita already gave us a nice introduction to the title of our presentation today, so I'll go ahead and skip this slide. And here's what we will cover. So we're going to give an overview of the polymeric excipients within our portfolio, not only cellulosics, but also seaweed-based excipients, as you'll see. We'll talk about key material functional attributes. We'll narrow in on a couple of the excipients that we'll overview. And we'll talk about key functional attributes that are common between those two excipients, such as particle morphology. We'll talk a bit about moisture content. And then we'll kind of diverge and look at the excipients independently and what functional attributes are important to each one of those excipients in their particular application space. And then finally, we'll wrap up with some of the work that we're doing around data analytics to improve consistency and performance. So this is the slide I was talking about. This is the overview slide. You can see there are a number of polymeric excipients within our combined portfolio, broadly based upon cellulose as the backbone or seaweed as the backbone. But we also have non-cellulosics and non-seaweed polymeric excipients such as polyox in our portfolio. Now, what we will zero in on today in our presentation is Avicel PH. So Yeli will talk at length about Avicel PH, as well as Methicel HPMC, Hydroxypropyl Methylcellulose. So I'll speak to that. And on the next slide you see here, just basically, I mean, obviously there are many more, intricacies, if you will, in the production of these polymeric excipients. But you can kind of see the broad overview here of, we start from wood pulp. We go through, and what we receive are these wood pulp cellulose rolls, if you will, and then we take those and convert them essentially through a number of manufacturing operations into, at least in the two cases we'll talk about today, Avicel microcrystalline cellulose and Methicel hydroxypropyl methylcellulose. And then you see up here on the top, basically that starting material. So what we're talking about are anhydroglucose units over and over and over again that make up this cellulose backbone. And this will come in handy later on in the presentation, but you can see that on each one of these anhydroglucose units, there are 3 hydroxyl groups, and those are at the 2, 3, and 6 positions on that anhydroglucose unit. So just keep that in mind as we move along in the presentation. And then here on this slide, what we're showing are basically functional attributes that are common between the two of them, kind of in that overlapping regime, such as particle size, particle shape, porosity, surface area. By the way, this list is not exhaustive. It's really kind of those key ones that we see more commonly, if you will, in our everyday operations. And then you see kind of the functional attributes that are specific to microcrystalline cellulose, such as plasticity, hydrogen bonding sites, water binding capacity, pulp type, and those that are specific to hydroxypropyl methylcellulose or HPMC, such as molecular weight, which our proxy for that is 2% viscosity, as well as substitution, and we'll speak more to substitution as we move along in the presentation. And so at this point, I'd like to turn the presentation over to Yeli. Yeli, how about particle size, and how is that important to the functional attributes that microcrystalline cellulose imparts into the formulation?
So let's start with particle size of MCC. As we know, particle size and shape are critical to MCC's flow property, and it actually also impacts its compactibility. For example, here you see different grades of Avicel PH. They have different particle size. From the SEM image from top to bottom, you see like Avicel PH 1.5, move to Avicel PH 1.1, and then to left to Avicel PH 1.2, and finally Avicel PH 200. The particle size gradually increase. Also, the particle shape become more spherical. As particle size increase and particle shape become more spherical, it gives powder actually better flow property. But on the other side, you may observe a slight decrease in its compactibility when they be used in formulation.
Yeli, before you leave that slide, that's interesting that you bring up flowability and compactibility. At what point between these different grades would you say you kind of switch from a granulation type of a grade to a direct compression grade, for example?
Typically, customers use Avicel PH 1.1, more like in the wet granulation type. It doesn't require much of particle flow, good flow property. And Avicel PH 1.2 is a common grade used a lot in direct compression because it gives a decent good flow property. And if your formulation really have a flow issue, then we can help customer with Avicel PH 200 because it can really boost up the flowability of the formulation. So
okay,
you can see a general guideline will be between Avicel 101 used in wet granulation type of process, and Avicel 102, pH 102 using direct compression.
Thanks.
And so how actually you utilize the coarse grade to benefit the formulation, right? So we know coarse MCC can improve formulation flowability. Here's the example how it actually helps. Compared to Avicel PH 102, the larger particle size and more spherical particle shape of Avicel PH 200 improve the flow property of the formulation and provide more consistent dye filling. So you can observe a quick stabilization of compression force and also a consistent and increased tablet weight, right? On the left side shows a consistent compression force, and on the right side you're going to see a more consistent tablet weight. So, well, on the other hand, you can also use smaller particle size to benefit, improve formulation. For example, here I compare example of Avicel PH 105 versus Avicel PH 102. So Avicel PH 105, remember, it's a smaller particle size grade MCC, and the Avicel PH 102 is compared to PH 105, it's a slightly bigger particle size grade MCC. So typically, Avicel PH 105 provides better compactibility in formulation because it provides larger surface area, right? When you compress it, it gives you more surface area to give you more binding site. It basically has a higher dilution potential. That's why it provides stronger tablets. So, for example, if you have a formulation that needs to improve tablet hardness, you might want to replace a small portion of Avicel PH 102 with Avicel PH 105 to improve the overall formulation compactibility and eventually, of course, a stronger tablet hardness. So I think too, I know particle size and shape also play an important role to HPMC's performance.
Yeah, good question and good lead-in, Yeli, to this section for me, right? So when I think of particle size, so let's really talk about the aspect of modified release, right? So what I'm talking about here are modified release matrix tablets. And I'm not a great artist, but if you look at this cartoon that I tried to draw here on the right-hand side of this slide, the little spheres are supposed to represent HPMC particles, right? And so I like to think of this is a fundamental, foundational, base of the pyramid type of a thing, if you will, when it comes to achieving robust modified release performance. What I like to think of is I like to have a percolating network, kind of a uniform lattice, if you will, of the HPMC particles embedded throughout the entire matrix tablet. This is called the percolation threshold, right, for a number of folks who are familiar with that term. Percolation threshold is impacted by really two factors, or other things, right, but two primary factors. There's the particle size aspect, and there's the concentration aspect. So with a finer particle size, you can get away with a little bit lower concentration of HPMC in your formulation. The coarser the particle size, you might need a little bit higher concentration of HPMC in your formulation to be able to achieve this percolating network of particles throughout the matrix tablet. And why is this so important, right? So when this tablet is administered, on the right-hand side, one of my teammates, I leveraged this slide from Matthias Kunar, one of my teammates in Germany. But once this is administered, the tablet is swallowed, hits the gastrointestinal or the gastric media first of the stomach. That is an aqueous media. And those particles of HPMC that are on the outermost surface of that matrix tablet, they begin to hydrate. They begin to swell, and eventually they coalesce together to form this swollen polymer hydrogel, if you will, this matrix polymer hydrogel that envelops the matrix tablet core. And it's this matrix polymer hydrogel that's so critical for modulating the release of the drug from the tablet, okay? And again, I spoke to the fact that that HPMC is a derivatized cellulose, right? So on those 2, 3, and 6 positions that I talked about earlier, there could be a methyl ether. There could be a hydroxypropyl ether. So this is a water-soluble polymer, and that water solubility enables that hydrogel formation. But first and foremost, you really have to have that nice percolating uniform lattice of those particles throughout the matrix tablet. So thanks for that segue. As I go to the next slide here, well, particle size is not only important to robust modified release performance. As Yeli mentioned, particle size, regardless of whether we're talking about microcrystalline cellulose or in this instance hydroxypropyl methylcellulose, particle size can impact processability as well. And there are a number of ways to modulate processability of your hydroxypropyl methylcellulose, which can translate to modulating the processability of your overall formulation. You can manipulate the morphology of the HPMC particles, so you can make them more granular, more spheroidal. You can increase their particle size a little bit. Or you can use co-processing techniques, right? So you can keep the particles fine, but introduce glidants like silicon dioxide, for example, and co-process those together so that co-processed mixture imparts better processability, better flow. That's particle size in a nutshell, right? I mean, there are several other aspects related to particle size and particle morphology, for that matter, that are critical to performance, to processability, but there are also other aspects, such as moisture, that we'll talk about. And Yeli, how about moisture? Can you talk to us about moisture and how that's important from a functional standpoint regarding microcrystalline cellulose?
Yes. Besides particle morphology, moisture content is another very important critical material attribute for MCC. I'm sure it's also play an important part for HPMC. For MCC, the moisture content actually impacted many performance. First, it impacts MCC's compactibility. Typically, with higher moisture content, it will give you a higher compactibility. And the second, moisture content also impacts MCC's water activity, which eventually actually can influence the drug product stability. Since moisture is act as a plasticizer, so the reason moisture content can improve the compactibility of MCC is because it acts as a plasticizer. It increases the plastic deformation of MCC, and it also facilitates hydrogen bond formation. So that's why, like when you have higher moisture content, it will help produce tablet stronger. So here shows on the graph on the left a regular grade of MCC and its corresponding low moisture grade MCC. So, for example, Avicel PH 112 is a low moisture grade of Avicel PH 102, and Avicel PH 200 LM is a low moisture grade of Avicel PH 200. So everything else is equivalent. The only difference is moisture content. And you can see the lower moisture grade have a slightly lower compactibility than the regular grades. Right? And in addition, actually, the external moisture, like the environment or humidity, can also impact MCC's compactibility. So if you look at the graph on the right, you can see the higher humidity typically lead to a higher compactibility, and which means a stronger tablet.
Question for you. So, obviously you were talking more about moisture, right, and the impact of moisture, low moisture grades versus non-low moisture grades. But I remember what you were talking about earlier in the presentation about the impacts of particle size. And at least within this regime, as you go from a pH 102 to a pH 200, it doesn't seem like you're seeing that much of a drop-off, if any at all, in compactibility between these two grades. Would that be a fair statement?
Yes, sure. Actually, that's a very good question. When you test them like 100% pure, so this graph is just by itself, the material MCC by itself. So the surface area doesn't matter too much, right? So all the MCC surrounded by themselves. So as soon as they contact under compression, they have plenty of surface area to bond. But when you put them in formulation, that's where the difference starts to show up. The smaller particle size is going to give you a better compactibility because it has a better, higher surface area, right? So in a formulation, when you have diluted with other particles surrounding the MCC, that's where the particle size and the surface area start to play. So typically with a smaller surface Avicel grade, you're going to have a stronger binding capability, that higher dilution potential because it has a higher surface area.
Yep, that makes sense. And sorry, another question. I mean, this is really interesting. So you talked about low moisture grades and non-low moisture grades. I mean, just out of curiosity, what's the difference there in terms of moisture content between non-low moisture grades and low moisture grades?
So I give you example, like 102, Avicel 102, that's our regular grade. The moisture upper limit is 5%, but typically you're going to see between 3 to 4%. And the Avicel PH 112, the upper limit is 1.5%. So a lot of times you're going to see right below 1%. So that's the difference. So it really gives you a better starting point, especially for moisture sensitive API, right? You're looking for excipient with a lower moisture content to minimize the interaction.
Yeah, I see. Thanks.
There are certainly benefits of using low moisture MCC. For example, if you have a moisture sensitive formulation, you might want to start with a low moisture MCC grade, such as Avicel PH 112 or Avicel PH 200 LM. Or there's a couple more low moisture Avicel grades available. The water activity of this low moisture MCC is lower than regular MCC grade. And this can help to minimize the moisture interaction with API and increase drug product stability. Imagine if you have a formulation with API, right, very sensitive to moisture degradation or interaction. So you want to start with excipient, which has a lower water moisture content to begin with, and then lower water activity. So I think this is definitely, moisture is definitely a big factor for MCC. It impacts it. You can see compactibility and also drug product stability. So Chu, how about the moisture impact for HPMC? I'm sure it's also a very important parameter, right?
That's right, Yeli, it is. In fact, I mean, I've worked on a couple of instances, right, where we've had, issues with tablet hardness and things like this. Because just like what you were talking about with microcrystalline cellulose, sometimes with HPMC, I think we have a similar, kind of limit there around 5%, no more than 5% moisture, right? But sometimes for some customers, that moisture level is really important to prevent things like capping or delaminating or whatnot, right? And the other thing I'll mention, right, is some of our manufacturing capabilities, some of our manufacturing trains do have the ability, when we're manufacturing HPMC, to tailor, the moisture level. We want to remain low, right, so that we can deliver a stable product that's not going to have, stability issues or compatibility issues with drugs that may be susceptible to moisture and things like that. But on some of our trains, we do have the capability to dial that in when customers so need it. And so thanks for that segue. So I'll move into the next slide, which is one of the slides around HPMC. And you may be wondering, right, the folks that are tuned in, you may be wondering why we're asking each other so many questions. For example, some of this work that I'll now present, this was conducted about 10 years ago, when, Yeli's organization and my organization were not yet a single, organization. And so I can tell you there are a number of instances and a number of formulations where I have used Avicel microcrystalline cellulose. I really like that aspect of compactibility and processability that Avicel microcrystalline cellulose brings into some of the formulations that I'm working on, that I'm developing. Other times I haven't used it, right? So in this instance here, I'll show this example A. Here we didn't use, Avicel. We used lactose. For example, this was a granulation that we had worked on. So we're still learning from each other. We've been together as an organization here for going on 2 years. So it's really good to kind of learn from each other more and more as we grow together here, and this organization is now one. So let's talk a little bit, about this paracetamol or acetaminophen example. And here, again, we're going to switch back to hydroxypropyl methylcellulose. Just kind of keep a mental snapshot of this formulation in your mind, right? So there's nothing really complex about it. It's acetaminophen 50%. It's Methicel HPMC, K4M 30%. There's some lactose in there as a granulation aid. There's some lubricant in there as magnesium stearate. There's some talc in there as glidant. But you can see here, again, nothing very sophisticated about this formulation. But it comes in handy as I move through my next few slides here. And I'll talk to more specific functional attributes to HPMC in this section, right? So things like polymer molecular weight, which we measure as 2% viscosity. Substitution, as I mentioned, HPMC is a water-soluble cellulose derivative, right? Some of those 2, 3, and 6 positions have been derivatized with methyl ethers and with hydroxypropyl ethers. So we'll speak to the importance of that as we move along here. Well, first let's talk about molecular weight, and I kind of like to use, maybe it's oversimplifying a little bit, but I like to use the microscope analogy. You have a coarse knob that you can dial in, and you have a fine knob that you can dial in, right, when you're really trying to resolve that image that you're looking at on the microscope slide. I kind of like to think of 2% viscosity or molecular weight of the polymer as the coarse knob. Right, so you can see here, I'm not going to try to play this movie because usually that doesn't work too well in internet platforms, but what we did was we took a couple of still frames here. We took a time zero, if you will, if you see the picture in the middle on the left. Those are essentially the tablets right after they have been placed into the dissolution media, and we've just taken a picture of it. And then the picture right below it, you see those tablets after 24 hours. And you see, for example, on the right-hand side, the tablets that are containing Methicel K100LV, or the lower molecular weight grade, those are completely eroded away within that 24-hour period. And then you see a rank order to the left of that as you go from low to mid to high molecular weight grade. So next in line would be the K4M. You see that after 24 hours, there's obviously some of the matrix tablet remaining. There's more of the matrix tablet remaining with K15M in the formulation, and then finally, at least in this case, there's even more of the matrix tablet remaining after 24 hours when Methicel K100M is present in the formulation. And then, these are two different examples. Example B has a more poorly soluble type of an API that's more dominated by erosion mechanisms of release. And then you see on the right-hand side example C, which is yet another type of an API that's not quite as poorly soluble as that one in example B. But you get the drift, right? You see here with increasing viscosity grade or increasing molecular weight grade on the right-hand side, you go from, essentially an immediate release type of a profile with the light blue to an intermediate type of a modified release profile with the magenta or the pink. And then, as you go higher and higher with your molecular weight grade of HPMC, you slow that release down more and more. And so let's go back to example A here. What we did, I had the privilege of working with some really brilliant polymer chemists over in Germany. I had the chance to work actually in Europe for a couple of years, and I really became friends and have some great friends that I still work with to this day at one of our German sites. And I mean, we're really talking, brilliant polymer chemists. So I had the chance to start collaborating with them when I was based out of Europe. And one of the things we did was most of my everyday work was around modified release matrix tablets. So I was in more of a tech service role at that time. And we would get questions from customers, can you help me understand the impact of your functionality related characteristics of your polymer on the resulting performance of my product. And so we picked a polymer, we picked Methocell K4M, and over the course of about 3 years, we collected about 20 batches or samples from 20 commercial batches that were produced in our commercial manufacturing facility. We tried to kind of, look and design in, if you will, and screen for as much variability from batch to batch in those functionality-related characteristics as we could find. But in the end, we produce very consistent product, at least in our commercial facility. And that's really what I try to depict in this slide as an intro, a further intro to example A. That graph on the left-hand side, just, in a nutshell, what we're talking about here is, these polymer chemists I was referring to, they have the ability in the R&D lab or in the pilot's facility to design in more substitution heterogeneity along that cellulose backbone, right? And those are kind of those red points that you see there in that graph. And then our 20 commercial batches of our samples from commercial batches of Methocell K4M, those are the green dots that are kind of essentially right on top of each other in that lower left-hand graph. So it kind of gives you an idea of really what we're talking about in terms of grand scheme of things, variability from batch to batch of these 20 commercial K4M materials. On this slide, what we essentially did, so think back to that that formulation, that paracetamol formulation, example A, right? We had 20 different commercial K4M batches. We had this very non-sophisticated paracetamol matrix tablet formulation. And what we did was we kept that formulation constant. We kept that as our constant. And then we plugged into that formulation these 20 different Methocell K4M materials that I've been talking about. And then we subjected them to dissolution testing, and you can see here on the left-hand side you have essentially 20 modified release profiles that are, for all intents and purposes, at first glance, right on top of each other. Okay, we did our best to minimize measurement variability. For those who have ever developed dissolution methodology with hydrophilic matrices, they can kind of stick to the surface, they can float. So we basically minimize that measurement variability ourselves as much as possible using sinkers, using hanging baskets, these sorts of things. And these are the resulting 20 modified release profiles. Now, if you look towards the latter part of that 24-hour release profile, you start to see that those curves are fanning out apart from each other. And so we zeroed in on that, and we did some very simplistic linear modeling and jump statistical software. And we found that there is a rank order, at least with this formulation and in this case, to those functionality-related characteristics. We found that hydroxypropyl substitution, for example, is a statistically significant factor dictating modified release performance. The molecular weight grade, or the 2% viscosity, is of borderline statistical significance. And then we found that particle size, all of these 20 batches met our CR-type particle size specifications. So in this case, that was a non-statistically significant factor, and methyl substitution was also a non-statistically significant factor, just because across those 20 batches, the methyl substitution was so tightly controlled. And then when you look at those, break it down and look at one of the time points at 22 hours, for example, the percent dissolved at 22 hours, and you look at that against those two most statistically significant factors, hydroxypropyl substitution. In the graph on the left-hand side there, or well, on the right-hand side, there's two graphs, right? So that first graph A on the right-hand side, if you will, that's drug release at 22 hours versus HP, and then that's that B graph on the right-hand side is drug release at 22 hours versus 2% viscosity. And what you can see here, it lines up exactly with what we saw in the linear modeling. There is a pretty clear relationship between drug release at 22 hours and hydroxypropyl substitution. As we increase HP, we increase the amount of drug that was dissolved at 22 hours. As we increase molecular weight, or 2% viscosity, we decrease the amount that was dissolved within 22 hours. But you can see that the relationship between drug release and HP was much more defined, if you will, much stronger than that relationship between drug release, at least within the regime we're talking about in this study, between drug release and 2% viscosity. Now, I have to say, take it with a grain of salt, right? Because if you look at that y-axis, we're only talking about 79 to 86%. So we're really only talking about 6 or 7 percentage points here, right? And so I'll go back to the fact that these 20 batches did deliver very reproducible modified release performance. But if you really dig into the data, you can see that there are relationships there, statistically significant relationships.
So, yeah, so this is actually very interesting. So it's a very robust, consistent product, but also you have some parameter you can fine tune, right, to really further control the performance of the polymer and the final formulation. For MCC, actually, the beauty of MCC is that there are many critical material attributes. They work together in the formulation to make it work. Here listed just the 4 unique critical material attributes for MCC: plasticity, hydrogen bonding site, pulp type, and the water binding capacity. So all of them together, they contribute to a stronger tablet and an easier manufacture process across the board. Like from direct compression to wet granulation and dry granulation and other process. So that's the beauty of MCC. Many factors like critical material attributes work together. So the conversion of the cellulose pulp to MCC actually breaks many intermolecular hydrogen bonds, which allows MCC to go through a plastic deformation and form new hydrogen bond under pressure. So that's how it actually acts as a dry binder in tablet formulation. So for the plasticity of MCC and the multiple hydrogen bonding site, we know they are critical material attributes, right? They contribute to MCC compactibility. And as it shows here at the schematic drawing, during compression, the MCC goes under a quick plastic deformation. It creates a large contact surface area, as we talked before. And at the same time, many new hydrogen bonds are formed instantly among adjacent microfibers. That's how it gives a strong binding and provide a strong tablet harness. So, for example, if you compare MCC with lactose or dicalcium phosphate or starch excipient, those are like commonly say binders or fillers, right, in tablet formulation. You can clearly see MCC provide a much higher compactibility than the others, right? It's all this compactibility, high compactibility is actually contribute to its plastic deformation capability and also multiple hydrogen bonding site give like really a strong binding capability.
Another question for you, when you're showing the plastic versus brittle plastic. So, you've talked about different aspects here around microcrystalline cellulose. You've talked about particle size. You've talked about moisture. You've talked about hydrogen bonding. When I look at that, do those attributes that you're talking about, changes or manipulations in those attributes, do those change, the different Avicel grades from plastic to other types of, brittle plastic, for example, or is MCC MCC? So does it always give plastic deformation?
Yeah, MCC is MCC, so it's always a plastic deformed material. The fundamental won't change. Of course, there's slight difference, right? For example, higher moisture will increase plasticity a little bit further, right? But fundamental MCC is a plastic deformed material. So between different grades, it's a fundamental. It's the same chemistry and the same basic material property. It has a similar critical material attributes. It's very different from other like dicalcium phosphate or lactose or starch. But then there's slight difference within each grade.
Excellent, thank you.
So besides the binding site, right, plastic deformation, those critical material attributes, the pulp type is also another very important material attribute, especially in its application in granulation process. For example, if you compare Avicel PH 101 and Avicel PH 301, there are two different MCC grades that produced from different pulp type. So you can clearly see, right, there's a difference in their performance when they use in wet granulation process. For example, here on the graph on the top, it actually is the same formulation. It went through the same granulation condition parameters, and you will see you need less liquid binder to reach to the same granulation endpoint when you use Avicel's PH 301 than if you use like Avicel PH 101. In the meanwhile, after the granulation, the formulation that contains Avicel PH 301 will end up with a granule that have slightly higher bulk density. So pulp types do play a big role in granulation process. And another critical material attribute is water binding capacity. This is also very important, especially for wet granulation process. So here I show you an example of a case study. They evaluated 3 different Avicel pH samples. They are produced from different pulp, and so they have different water binding capacity, right, as you see on the graph on the left. And we compare them in a theophylline model formulation and use the extrusion fermentation process to make spherical pellets. So they all go through the same, use the same formulation and went through the same process condition. But they end up with different D50 and also different particle size distribution, which eventually lead to different yield, as you see from the bottom graph on the right. So they give you very different yield. At that time, we were targeting for 700 to 1,000 granule size. So you can see the water binding capacity is another very important critical material attribute for MCC. So overall, you will see all these material attributes are important. They directly definitely impact MCC performance. So therefore, a consistent MCC also beyond MCC, right? Excipient is very important. The consistency of the material attributes and the batch-to-batch consistency are very important. Because it do help customers to produce a robust formulation in the final product.
Yeah, Yeli. I mean, one word I really picked up on when you talked about this slide was consistency, right? And our customers do expect consistency from batch to batch. And so really that leads us into the concluding slides of our presentation. So for this last section, what we're really wanting to talk about here is, well, what are we doing, from a manufacturing standpoint to ensure that our customers are receiving as consistently performing and as consistent quality product as possible. And really that's the name of the game. And so here I just kind of have a depiction here. Actually, I leveraged this slide from our manufacturing and quality colleagues. But Yeli and I have had the privilege of being on a couple of teams here in the last couple of years, where if you go into one of our control rooms, you will now see a dashboard, right, up on the screen, where our manufacturing colleagues, when they're producing these products, are screening or are looking at these critical to quality and critical to performance attributes, and they can see there, are we within targeted ranges, right, for these attributes that may dictate compactibility, that may dictate modified release performance, et cetera, et cetera. Are we within target there? And do we need to make kind of any minor, adjustments within validated ranges, in situ, to make sure that we're always staying within target? Yeli and I also have the privilege of sitting in on another team. We call this, just for short, internally, our multivariate analysis, or our MVA team. But we do so much more than that as a part of this team, right? This team is led by one of our colleagues over in the UK, Mohammed Kadir. And we have now, to this point, and we've done this just over the last couple of years, we've to this point looked at a number of our polymeric excipients that are within our portfolio. In addition to the multivariate analysis, we've done univariate analysis. We've looked at aspects such as what we've talked about today: loss on drying, pH, particle size, substitution, molecular weight. We've done this for a number of different products. I think this PCA plot that you see here, if I'm remembering correctly, this is coming from, for example, Avicel PH 102. And so what we've done is we've looked at years worth of data. We've done univariate analysis. We've looked at our multivariate analysis as well, looked for relationships, and most importantly, we've armed ourselves with data so that we can go and have informative conversations with our customers who are taking these excipients and inserting them into their formulations, whether it's granulation type, unit operations, direct compression, whatever it might be. But our customers are very interested in looking for trends, looking for relationships, so that they and we can work together to understand, any relationships that we see. Do those translate to quality, or do they translate to performance attributes for those customers when they take our excipients and formulate them into their drug products? So that's just a little snapshot. Really, the possibilities are endless. We've taken you through a number of aspects. We've looked at microcrystalline cellulose, attributes that are specific to MCC. We've looked at HPMC, or hydroxypropyl methylcellulose, and attributes that are specific to HPMC. We've looked at attributes that are common to both of them. And then we've looked at, what are we doing from a digitalization standpoint and a data analytics standpoint to understand how we, can really deliver top-class consistency from batch to batch to batch for the customer. And with that, that takes us to the end of our presentation for today. Thanks for sticking with us. And I'd like to hand back over to our moderator, Rita, so that we can discuss any questions.
Well, thank you very much, True and Yeli, for that presentation, that information. Before we get started on the Q&A, just a quick reminder to our audience that they can submit questions by typing them in the Q&A box, which is located on the right-hand side of the presentation window. So, True, let's start with you with this one. On the modified release side, how would you rank order the critical attributes?
All right, yeah, thanks for the question, Rita, and whoever was asking it. Yeah, I like to think of this, in my mind at least, as kind of a pyramid approach, right? So first and foremost, I look for particle size. I think if you don't have, a percolating network throughout that matrix tablet, if we're talking about modified release in this case, right, if you don't have a percolating network of those HPMC particles throughout the HPMC matrix tablet, then that's kind of a non-starter, right? So particle size is number one for me. Then beyond that, I look at, the chemistry, right? The chemistry of the HPMC polymer in this case that goes into the matrix tablet. Most of the time it's going to be an HPMC 2208, which means 22% methyl substitution, 8% nominally hydroxypropyl substitution. That's what's most commonly used. Sometimes an E chemistry is used. Sometimes I've even seen a different chemistry, such as an F chemistry, for example. I look at that next. Then I look at molecular weight or viscosity grade as we measure it, right? Because that dictates the hydrogel resilience, if you will, chain entanglement of the polymer, the swollen matrix polymer hydrogel, and then chain disentanglement. And then finally, if we need to make those little minor tweaks, like what I presented there in example A, then we look within a chemistry, right? So there is room within the compendial box, if you will, for HPMC 2208 to play there in terms of substitution. So that would be kind of at the tip end of my pyramid, if you will. But that's the rank order I think about.
All right, thanks, Drew. Here's our next question. So what are the preferred analytical tools used during testing and production and tools used for particle analysis?
Good question. Yelly, you want to take that or you want me to?
You can. Please go ahead.
Okay. Well, I'll start, and then if there is anything to add, please, that I missed, please feel free to add it. Analytical tools. So I think there are many, right? I'll tell you there are analytical tools that we use in our QA/QC lab with regards to particle size, I think was the question, right, specifically. Things like percent through sieves. So if you get a C of A from us on, we have multiple, grades, if you will, of Methocell for modified release, but you may see a percent through 3 different types of sieves, like a 100 mesh or a 40 mesh maybe to start with a quartz sieve, then a 100 mesh, then a 230 mesh, for example. You'll see those. I will tell you on the R&D side, though, we use other types of particle characterization techniques as well. Things like particle imaging, quick pick, for example, is one that we use quite frequently. Laser light diffraction, I know a number of customers like laser light diffraction type techniques such as Malvern. So there are a number of different ones. I mean, we do have these, and we utilize these and leverage those to characterize particle size, particle shape in our laboratories, whether it's our QA/QC in the instance of sieve analysis or our more sophisticated type techniques that we use in the R&D lab, such as quick pick or laser light diffraction.
All right, thank you. Here's the next question. So do you look at any other parameters like porosity or surface area?
Ooh, good one. Yelly, you want to start there?
Yes, we do look at those. It's not our COA, so it's not a specification. That's not come with each lot, but we do look at those and closely look at those at R&D level, and also use it as a tool for troubleshooting. Because PET surface area, like a specific surface area, and the water binding capacity, those are actually also very important material attributes, especially for MCC, right? It's contribute to its functionality that customer is looking for. So we do look at those as our research tool and also as a troubleshooting to support customer.
All right, thank you. Here's the next question. So how do you determine water binding capacity for Avicel?
Water binding capacity, that's a test. Actually, we do have a test method we can share if we can take it offline, right? I don't want to go through the detail of the test method, but basically it gives you data, like value, to see, okay, what's the water binding capability this material has versus others. And this parameter is particularly useful for if you use it in wet granulation process, because you're actually adding liquid, so the water binding capacity is very important because it impacts how you're going to reach the end point. So this one, like the test method detail, if you're interested, we can take it offline.
All right, thank you. Next question. So what if MCC alone can't improve flow and compactibility issues? Are there other options?
Sure, yeah, this is actually a very good question. Yeah, like many materials, right, MCC has its limitation, and there are many options actually to further improve its flow or compactibility properties. One very effective and feasible approach to achieve better functionality that typically excipient companies use is through co-processing, like to produce a higher, like multi or higher functional MCC materials. I can give you a couple examples. Like Avicel PH is a co-processed material with 98% of MCC and only 2% of silicon dioxide. And this material has improved flow, right, and also compactibility when you compare to pure MCC. Another good example is Avicel DG, which is actually co-processed material with 75% MCC and 25% dicalcium phosphate. So this material provides actually better compactibility and also better recompactibility. So it's because of the introduction of the second small portion of the brittle material, dicalcium phosphate. So the combination of plastic deformation and brittleness coming from the DCP actually really contribute further to improve its compactibility. So yes, so that's co-processing is one way to actually really give additional functionality to MCC. Hope this answers your question.
All right, thank you. Here's another one. So what are the main components of your digital analytics capabilities?
Oh, good question. Yeah, we didn't have a lot of time to go into details there. Well, that could probably be a whole webinar in and of itself. So main components, right? So we, kind of behind the scenes, we'll have this team that function, that they'll focus on univariate analysis using capabilities such as Minitab or JUMP statistical software. We have multivariate, as obviously we showed a snapshot there of our principal component analysis plot there. That's done using this software called SIMCA. So we'll have, basically a statistics expert as well on our team that can take the statistical analyses to the next level. So those are really kind of the key components as I see it in my mind when Yeli and I are sitting in on these meetings having these very in-depth technical discussions with the rest of our multivariate analysis teammates.
Okay, thank you. We have time for one more question. So do you have a brief explanation on F chemistry versus E chemistry?
Oh, yes, good question. I kind of blew right past that one, right? So these are basically just different substitution targets, if you will, of hydroxypropyl methylcellulose. Both are HPMC, just like K chemistry, which is 2208. I think I mentioned that one. So E chemistry would be 2910. So what does that mean? That's 29% nominally methyl substitution and then 10% nominally hydroxypropyl substitution. F chemistry would be 2906. What does that mean? That's 29% methyl substitution and 6% nominally hydroxypropyl substitution. So I guess if I had to, sum it up in a nutshell, F chemistry just has a little bit lower hydroxypropyl substitution nominally than does E chemistry.
All right. Well, thank you very much, Drew. Unfortunately, we are out of time, so I'd like to thank our audience for attending and participating in today's event. And I'd like to thank our sponsor, DuPont, for making today's webcast possible. You'll receive an email alerting you when this webcast will be available for replay, and I invite you to forward that announcement to your colleagues who may have missed today's live event. So thank you again for joining us, and have a great day.